Good evening, everyone. It's a privilege to be here this afternoon. I'll be talking on phylogenetic traits or working on phylogenetic traits. Please just give me a moment. Thank you. So my discussion is not going to be like talking and talking. We're going to be doing a lot of working together this afternoon, and I just hope that I'll be able to explain as little as I can. Like I will say, I'm also a learner, and that we're going to be learning together this evening. So we're going to be looking at phylogenetic traits. Why do we have to talk about phylogenetic traits? How do we do them? What are the tools that are available to draw a phylogenetic traits, and what are the approach we should adopt? The person that taught us, that just concluded, did a very great work on navigating the blast environment for identifying an unknown organism. And I think we are going to be building up from there because we are going to be doing some activity from there. So let me quickly do a quick introduction of what I'm going to do, what my work will look like. I just hope that the time allotted to me will be enough to be able to cover all of this, because there are two are together, but the last part is kind of another different thing, which may also need a lot of time and a lot of follow as we do it. But the first thing that we're going to be talking about is constructive phylogenetics using MEGA. But before I go into that, like somebody asking the question that, can you use other form of tool to draw phylogenetic trees? The first thing I'm going to tell you that it depends on what you are doing. Sometimes when you're working with, for example, I work with whole genome a lot, and when you're working with a lot of whole genome and you want to compare them, sometimes MEGA may not be too sufficient to because of its capacity. And then you could be trying some command line tools like IQ tree, and then there's another one. I think somebody mentioned split tree. There's another one. There are so many other ones, but I may not be able to recall all of them now. So, but MEGA is common. It's very easy to use because it's available on all interface. You can use it on Linux. You can use it on Mac. You can use it on Windows. So that's why MEGA is the commonest. There are other ones. There's something that is called I think it's Fig5. That one can also be used for phylogenetic tree. So those are the ones that we can use for phylogenetic tree. But then another thing is that after we've done our phylogenetic tree, we want to make it more beautiful. We want to make it more appealing. We want to do some coloring to it, which we cannot do with MEGA. MEGA is limited in aesthetics sometimes. When you're working with MEGA, sometimes when you're going to send your work for publishing, sometimes the journal house wants you to italize the scientific words, and then when you are going to do that on MEGA, that is not possible because you'll be italicing some of the strain names, which must not be italiced. So when we come to iTool, that is possible. So we'll be able to look at if we're able to run through that this afternoon, and then we'll work together. So, but one thing I want to say is that with iTool, we must have drawn our tree either with MEGA, with IQ tree, or with any other tool. There are many other tools that are available on. I think that there's a tool that you can work with R to draw phylogenetic tree, but then remember the tool that R uses also. So that you can use to get your tree file, which is usually new week, then you can bring it to iTool then for you to be able to visualize them. So that's one thing we'll do. Then the third one is submitting your data. When you got your data, you want to put it, journal house wants you to make your data available. Then we have to submit it to a database, which is NCBI database or EMBL, or the DDO, which is that for that, or is it DDO or DDGA, which is for Japan. So that is about what I'm going to do. So let's now look at why do we draw phylogenetic tree. The first thing is that what we do is that the one thing that we do in bioinformatics or in genomics actually is that you want to understand a relationship. And one thing we do is the first, like the person that told you said, you have a query sequence and we want to know what it is, then we do a blast. Then it gives us a list of what is close to it. Then we want to see where is it coming from? Has it branch from his family, or is it part of the family? Are they the same thing, or something is very about them? It's going to give us their genetic context, their evolutionary makeup. That's what phylogenetic is going to point out to us. It's going to tell us, okay, how are they related to one another? And then one of the things that one of the way in which we know that, okay, an organism has evolved a new characteristics and is maybe speciating already. So that is how we know about that. So that's what we do with phylogenetic tree. It shows us evolution relationship among organisms based on what is contained in their genetic content, which is the nucleotides that I think you must have talked about that yesterday. Now, one thing that I want us to also see is that you see that this what I show you is like the tree I'm showing you is just like a kind of phylogenetic tree, and it's showing us what organism, how they are related to one another. Now, for example, organism A and B are more related than organism C and D to A. B is closer to A than C is closer to A. C is closer to D than D is closer to B. So that is what we are trying to see. How related are they? And then we see this part shows where they are coming from. Now, what we do in phylogenetic tree is that when you are going to draw your tree, you are going to get an organism. When you do your blast, you already know the family of organisms that you are working with, because it tells you by your blast search. I mean, maybe you have just sequenced, you have worked in the lab, you have seen some phenotypes that you are looking for, you have seen some characteristics. Maybe you just even come across the organism accidentally, and it has a beautiful phenotype. Then you can send it for Sanger sequencing. And when you do that, you bring it, you bring up your file together, your chromatogram, then you extract your nucleotide sequence from it, then you do a blast or a blast search on it. And when you do that, it tells you what organism you have. Then what you do, like the facilitator that just finished has told us, we do a blast, then we download the one that of interest to us. So then when we do that, we know what's then we now look for the you see, for example, I work more with bacteria, and I'll be talking about bacteria. Now you will see that, okay, in this bacteria family, for example, maybe I got E. coli, I got Klebsiella, I got another group of Enterobacteriaceae, and I say, okay, oh, this is an Enterobacteriaceae, but the what I got is E. coli. It's showing me a lot of then I can go and bring out Klebsiella or Yersinia that is close to them, and then, okay, I'm using that as my house group. That will want to tell me, we want to show me how they have come from in their ancestry. So that's what we do. That's what phylogenetic is showing you. Another thing that we're going to be considering phylogenetic is that what we talked about, the probability of them being the same, and that's what we call the bootstrap percentage. We will be doing all of those all of those practically as we work. So I'm just giving an overview of what it is. So it tells us that, okay, this is percentage of how close they are, and then the higher that percentage, the more reliable our result is. So that is about bootstrap. I've talked about house group. I've talked about the what you have here, what you have at the tip here, which are your organisms, is what we call the leaves. Then the internal node is that one, this one that I painted 9 and 2, which was where they are coming from. Okay, and then the blue line shows the evolutionary length. Now, when we want to run phylogenetic tree, from this part, I'm going to change my class to how will I call it now? To practical oriented class. So I'm going to be switching over now, and I'll be explaining a few things as we work together. Now, but before we do that, all of us are working together this afternoon, sorry. It's morning where I am. So, or sorry, this evening. We are, all of us are working together now. I'm going to send a link in the chat where I don't really know. I should have done this earlier. I'll give that to you because it may take a lot of time for us to be able to do that. But I don't know, maybe we can just get to download Mega, or maybe if you have Mega, I'm sending the link so we can download Mega, we install it, and we can work around it. Maybe that will take our time. I can just show us from my examples, but I would have loved it if we can just work together. Let me send the link first, and as many people that can just install it, they can follow me as I try to demonstrate a few things. Just give me a moment, please. I'm sorry, please, just a moment. Okay. So I think we can download Mega here, and then we can work with me. But if not, I'll be working and I'll be showing us what I have to do. Now, the next thing I want us to do, everybody, is this. Now, I think there's a sequence. I'm going to be sending a sequence here again in chat, and that sequence we are going to just give me a moment, please. So I don't know, maybe we have this sequence already. I think this is the sequence that is being used throughout this training. And I want us to copy this sequence and do a blast search. And then I'll be doing the same thing. I'm going to show you what I have. Then we're going to download just a few. We're going to copy out a few nucleotides from there, which we want to use as our training guide. And I'll be explaining along the line as I work, as we work together on my screen on Mega. So please, if we have it, we can always do a blast, and then I will tell us what to select. So I'm going to be switching over to another screen now, so you could see what I want us to do. Okay, I guess we can see my screen now. So this is the blast I've done, and I want us to do that, and then I will continue I want to select the ones that I think is good for me to work with. I'm going to be selecting only 10 sequences or 10 genomes. All of them says this is 16S sequence, and I'm going to select them, and we're going to be working with them. Now, it's a very good thing that if you can see my screen, you will see that all of this says these are pseudomonas. They are pseudomonas gene, and then one of them is saying that it is rhizobium. I think let me count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. The 11th one says is rhizobium. So the first thing I want to do is, if you look at from what our teacher has told us, that the e-value is very, very important, and then it shows us that the e-value is 0 plus 0.0. So they are reliable sequence I can trust because of the e-value first. I can trust, okay, that these organisms are related. Then the next thing is that the query cover. Now, the query cover shows us that there's a lot of discrepancy, and then many nucleotides are not the same. That's the meaning. Many nucleotides are not the same, or the length. It depends. It could be many of the nucleotides are not the same, or the length is not the same. The length of the one I submitted with what is matching to, it's not the same. So that's the thing that we are seeing. That's why the query cover is kind of varied. But with the e-value, is something to be able to go with, because it's still within the range that we can trust. But although what we can trust before we can think, okay, they might likely become of the same species, is from 98% and above. But we can still just work with this for our class. So let's just download the FX10. So what I'm going to do is I'm going to unclick this place, and then I'm going to select my FX10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then, okay, 10. Then this one plus this one, okay, 11. So because he's saying this is rhizobium species from the scientific name, but the description that was given is Pseudomonas. So let's see let's see what is going to show us when we draw the tree. Now, one thing I want to also see is that if you notice that when you look at the scientific name, they have different species in some of them. For example, the first one say originosa, the second one, the next one, the fifth one say species, then the next one say Guisenia, and then the other one. So we saw that they are likely of different species. We may not be able to conclude this. So, but what we want to do now is download this, and then we are taking it to Mega. That's where our work will start from. So then I will start to talk about that from Mega. Now I've selected my 10, then I think we will come to this place. You see, it said download faster complete sequence. That's what I'm downloading, because it's already 16S. That's what I'm downloading. And that thing I want to mention is that sometimes when we're working with 16S, the sequence you gave might go and align with a particular region in a complete genome. So if you don't see 16S in this place, please make sure that you are careful to select. If not, if you are going to download, it might download complete genome, which could be a lot of length bigger than what you're expecting to work with, and that might affect your work on Mega. So that's why I try to point this out to us. So the next thing I want to do now is I'm downloading this. Look, I have many options here. I have fast A complete sequence, fast A align, gene bank, but what I'm interested in is fast A complete sequence. I don't want the align sequence alone. So let's copy everything, because I'm taking to Mega, which will still do that for me, and we will seek that out, sort that out for me. So let's do that now. Let's just download it. Okay, so it gave me a name. Now, it is imperative for me to rename this and save it as .faster, because Mega will work with .faster. So let me rename it. Let me just say the Pseudomonas training .faster or fast A. Then I change this to all fine. Okay. So the next thing, now I've saved it. Now in this place I should have 11 sequence. So the next thing I'm going to do now is I am going to Mega. Now that's where I will be explaining that's my PowerPoint now. I'll be talking. I'm going to be moving around, but I just pray that we don't get confused as I move around. Before I move to Mega, I want us to see something in that what do you call it, in my PowerPoint again. So let's just go there. Now I'm moving. I want to share with us something in my PowerPoint, and I want to explain some things quickly. Okay. Now,
excuse me, sir.
Yes, are you with me?
Yeah, it appears your screen is frozen. We are still on the National Library of Medicine. We didn't see how you renamed faster.
Okay, I'm going to. Oh, you didn't say I did what?
How you renamed faster?
Oh my God, I'm so sorry. Okay, let me try to do that again. So let me move this away. I'm sorry. Please, I also like to ask. I hope I'm not going too fast, because I know I can speak very fast sometimes. Hello, I want response. Okay, thank you. I'm not going too fast. Thank you. Okay, so please, I'm going to be switching over now, so you see. Oh, I think I'm here. You were okay. It's my fault. Okay. How will I do that? Because that will be on my. Okay, let me see how I can do it. Just a moment, please. Okay. Okay, just a moment now. So let me go. I'm just going to show us how I rename faster now. I'm showing this place. This is why I rename faster. Okay. Now this is. Let me see. I'm coming to download again, so you will see what I downloaded. Then I think you'll be able to see where I'm going to be saving it. I hope you can see it. Just a moment. Okay, I think I'm seeing it. That's good. Okay. Okay, I copy all of this. Then it says save. Can we see my screen now where I want to save? Are you seeing it?
Yes, we can.
Okay, so now this is, it gave me sec.dump. So I'm going to rename it now to, let me say Pseudomonas training file, Pseudomonas training file, Pseudomonas training faster. Then I change this to all file. Okay, so you see
Hold on,
I have
sir.
it now. Okay.
Sir, we can only see your file explorer.
Holy Ghost.
Yeah, that's what we can see. I guess you are sharing a particular window.
Yeah, I'm not sure. I had to share everything. I don't know why it's just giving me this issue. Okay, let me see. Let me see if I can share this. Just a moment. I expect it to show because where it is where I'm working on, I'm showing. And I'm seeing it moving around, but I don't know why it's not working. Because I can also see my screen from here. But I don't know. I can see that you are not seeing what I'm doing.
Yeah.
Okay, let me see. Let me see what I can do again, please. Just a moment, please.
Are you using multiple screens, sir?
Well, I can barely hear you. What do you say?
Multiple screens. Do you use any screen?
Yes, I'm trying to use all the screen, but it's not allowing me to do that.
This is the issue because you are using the screen.
Okay. Now, can you see all of my screen? This is fine. I think you should be able to see what I'm doing now. If I move around, you should be able to see what I'm doing. So let me just go back to this. Okay, can you see it now?
Yes.
Okay, now let's see. So let's just work with this now. So, for example, I'm going to download this. I already have that. Can we see this now?
Yes, we can.
Okay, now. So I'm going to be renaming this now. Let me call it, I'm calling it Pseudomonas training faster. So I have this before. Now, just to show us how I changed the name, because why I changed the name is because to give me a description of what file it is. So the next thing is now I've changed it now and I'm going to save it. So I know what it is, I know what I want to work with. So now the next thing we have to do is now to go to my mega file, to my PowerPoint. We'll go back to my PowerPoint. Can I see my PowerPoint now? Hello? Okay,
yes,
thank
you. Can
you.
see your PowerPoint.
Yeah, everything
Thank
is
you.
okay.
So what I want to say is this. Now I'm going to be drawing my trees from here, but I want to explain this. Now I can use, you see, when on Mega, there are many phylogenetic drawing tools, which we call them algorithm. They are based on different parameters. Let me use that word. For example, we have neighbor joining, we have maximum parsimony, we have maximum likelihood. Now, there are different reasons why we use each of them. Now, if I want to quickly look at what is the relationship between them, and particularly when I have a lot of data, for example, maybe my file has something like 500 FASTA file I want to work with, then it is better to work with neighbor joining. But what about neighbor joining is that, like I said there, it is less statistically rigorous. What it just is, it match the nucleotide distance by distance, and it's always very fast. That's what it does. But unlike maximum likelihood is, we used to say that it's assigned evolutionary, the evolution itself. It can tell you where they are come from. It can tell you the ancestral sequence, the way it works. So this one is kind of slow, and it is what, if you are trying to publish your work, it's what most journal want to see, because it's more rigorous, it's more statistically rigorous, and it's go beyond just align them distance by distance. It use other algorithms. So that's why maximum likelihood is the most commonly used. Now, and then the next one is maximum parsimony. Maximum parsimony, it's it's also fast, just like neighbor joining. But one thing about it is that it's only give us, it's not as thorough as the maximum likelihood. And then when they're having a lot of branches, there might be kind of confusion. That's why most articles want us to use maximum likelihood. Although it is also at the level of 16S, neighbor joining is often accepted. But if you are working with whole genome sequence, you have to, most of them will tell you maximum likelihood is the best that they want. So now, this one is not for our class, but I just put it, this one is just for, mostly this is used for population genetics. When you are doing population genetics, we use this Bayesian inference. And then first you will need some other tools to be able to work with it, which is this Mr. Bayes or Beast, to be able to use Bayesian inference as our tool on Mega. So it's better we stick to these three. So the next thing we are going to do now is, I want to now move back to Mega. Just a moment, please. Escape. Okay. So now, can we see my Mega screen? So this Mega, maybe for those of us who have downloaded Mega by now, we'll see what I want to do. So I'm going to call out my file. I come here, I click on File, then open a file session. So I'm opening the file I want to work with. So the next thing I do now is I'm going to look for my folder, which is this, and then I'm working with this. Now in this place, I have all of the files I've downloaded with their names. Then one thing we can do is when we get to Mega, we can trim them. So let me say open. Okay. Now the way see what it gave me two options. It says align or analyze. Then the next I click is align. Okay, do we see it now? Can we see my screen, please?
Yes, we can, sir.
Okay, thank you. So now, I'm going to do an alignment for this. So the first thing I do is that, so it gave me, this is what it gave me. It gave me, what can I call this now? This is their ascension number. So I don't know what organism they are. I may have to go and look for this, or let me see, maybe it's continue. Okay, no. So I may have to go and look for what is their name and then fix it later. But let's just work with this, and then we can be talking about how we will assign their names. So the next thing I want to do is now is that like the teacher that the facilitator that just finished taught us, she said we want to align them. In this case, now you could have aligned whatever you are putting in here with cluster omega or cluster W if you want to, because those two are also helped in Mega. They also helped in Mega, so you could be able to choose the one you want to work with. So, but in case you have not done the alignment before, the next thing we'll do is I'm going to select all. Just put your this, I said Control A. I've selected all. And the next is go, you click here, it says come to what? Alignment. Do we see that? Then we want to align them. Why do we align them? We want to put them in order, line by line, how they match one another, so that it will be easy for our algorithm to draw the tree. Okay, so the next thing we do is align by cluster W. I love cluster W a lot, and that's why I use cluster W. You could use the other one. What do I see there? Let me see the alignment tool again. Let me close this so that we can see the other one. Okay, muzzle. Yes, I remember. Sometimes I use muzzle when I have bulky number of organisms, because muzzle can still tolerate that sometimes cluster W fails. So that's why I use cluster W. So the next thing we do is align with cluster W. Then I come here, I click OK. I'm using default. It's better to use the default parameter as a beginner until you understand better how these things work. So I use default, and then it's going to do the alignment for me. Now it has aligned this, and then with this I can choose to draw my tree. But there's something I want to mention before I continue, which is going to take me to my next slide. Okay, sorry, to my slide, which is my next slide. Now is this. Just a moment, please. Now, you see, do you see this, my slide? Now, for what we are doing now, it may be needed or it may not be needed. But I'm going to explain to us that when we are going to be establishing during the phylogeny, we are going to a place that will tell us to ask us to choose test of phylogeny, which is called model, the model we are going to use. Now, those models we are going to be using could be any of these: Jukato, Kimutato parameter, Tamura, or GTR. Now, one thing is that all of these have where they are useful. This one work with if there's equal substitution rate between the nucleotides of the genomes, or maybe there's in this place now, maybe there's a change in some nucleotide. Maybe we used to have, for example, now it's going to be ATG, and then instead of A now it has changed to AUG. There's a transversion. Then we work with Kimuta parameter. So this take care of that if there's something like that in between the nucleotide or the protein we are working with. So that's why we use Kimuta parameter too. And then this one, if all of them have independent substitution rate, we use Tamura Ni. And GTR combine all of these, and then they can work together. But what I want to say is that whatever you want to use among all of them, the most important thing is that we go to Mega and first of all test our sequence. I didn't do that here because it's just 16 that we are working with. We first of all test the FASTA file that we have. It will tell us the model we can use. I'm going back to my Omega, to my Mega now. Let me do this. I'm going to show you something there on Mega. Mega now. Let me see if I can find that. I want to show something on Mega. Oh, sorry. Okay, let's continue with this. When I can later show that. So now I have this. Then the next thing I come here is I want to draw my phylogenetic. You say phylogenetic analysis. Do we see this? So once I click on this, the next thing it's going to do is it asks me protein sequence coding. Just protein coding nucleotide sequence. Click yes. So it's done that for me. Now you see it has taken me from that place to another place, which is this one. Now I can test my model to know which one will be the best for me to select when I'm working with, when I'm going to draw my phylogenetic tree. Now I come to this that they call model. Then we click on it. So it said find the best DNA to protein model for this. So it's going to tell me which one I'm going to use. So it said I should use maximum likelihood, branch swap none, then number of threads. Click OK. So this is what he has selected for us, and it's doing that for me already. So once he finished with this, the next I'm going to do is to draw my phylogeny. It's finding the model for me. So which of them is telling me, okay, which of the model will be good for me? It's giving me just for all of them, this will be the best model I should use. So the next thing is that in all of this model, it gave me for 20 of them, Kimura 2 will be the best. So it just gave me the best fit, the one that I can use. So what I'm going to do now is I know any of this model will work fine for me. So what do I do? The next thing is come to my phylogeny and say, I want to draw my phylogeny. Constructing with, let's work first with neighbor joining, and then we'll try with maximum likelihood. I'm going to try the two. First, neighbor joining. I told us neighbor joining is very fast. We just click on this, then click OK. Now let's look at it. So the next thing is he has selected the model for me, and then I will go straight to working with it. So this is I said, look at it, my bootstrap replicate. Repeat it 1,000 times. This bootstrap value means that I say 1,000 is mean that search round 1,000 times and see that for every 1,000 that you have searched this thing, this and this and this will be together. That's what the bootstrap is trying to say. So now test of phylogeny bootstrap repeat method. Then we click OK, and then it will start to run it for us. So my tree will be ready. So I have my tree already, and this is it. So you see, so this guy and this guy seems to be closer. This guy seems to be closer, and then this is maximum phylogeny. What about maximum phylogeny is that it shows us the one that is farthest, where they have likely come from. Now, another thing I want to say in this place is this. Now, look at all of these values. This value that you have here, 71, 75, 99, 99, 83, 99, 60, 36. Now, this thing that we have here are the test of phylogeny. Out of 100%, after moving around 1,000 times, 71% says that this and these two guys will be together. Oh, this one, 36 and 35, they are likely, they have evolved from the same ancestor. That's what it's saying. And then 75% say that these two are related to this. Then 99% say that they have come from the same ancestor, possibly. Okay? And then the same thing with the ball. Look at this guy. This 36% is telling us this and this, they have come from the, but 36 is very low. Anything less than 50% is not true. Okay? And so this is not true for this tree. So it's not true. These guys are not family. For example, if you are going to be saying that, okay, possibly they might have been the same species, or no, this is because they cannot, it just group them together because it has nowhere to place it. Because 36 is far way less than 50%. And that 50% is not too good to be considered from the same ancestors. There are so many things that are not related between them. So that's about this. Now, let's check this is the original, how the bootstrap consensus tree will look like, which is this. So these are the bootstrap consensus tree will look like. And then this guy is coming out. I don't know. Let me see. Maybe this is the guy that I told you that they were never the same. So you see that this guy is coming on his own. He's not staying with anybody. But in the original tree, you see OP87 and OP7261914. I think those are the ones that we have here. Do you see it? So I told us that they are never the same. And you can see that this guy is coming independently on his own, and this other guy too. So it just gives them that value to bring them together, just because there's something that must be there. But all of these one are we can reliably say that they are from the same ancestor as you see those bootstrap value. But say, okay, how related they've been. That's about that. Now, what next do we have to do? I told us that if you want to beautify our tree, now with this also we can do a lot of manipulation. Now, for example, maybe in all of this, let's say this is the one that is kind of far away from them, that guy that I highlighted. Now, what I want to do is I want to now make it the root sequence. The root sequence, what is that? That means I want to believe that this guy, because it's far away from them, it will show me how other ones has evolved. Then I come to this place, I click on this subtree, then I said, make it my root sequence. That's just it. So it's now the root, then it tells me how they have evolved with time. Possibly these two guys, they are independent on their own. And maybe this ancestor, this came from it earlier than the other, and then this is how they have come over time in relation to this guy. Do we understand? Do we get what I'm saying? So that is making this the root. And then I can also beautify this, color it here also in Mega. I can do that. I can give them different color because I want to differentiate them. For example, I want to give this guy a different color. This guy, this leaf OP8726, I want to give it a different color so that I will be able to point out that that's my root sequence. And then let's just do it now. Root, instead of, I already said it's root sequence. I want to show people that it's root. Then I come to draw option. Do we see that? Then say format subtree. So we come to this place, and I'm going to be formatting my tree. Now I'm not working on any of this. I'm not going to touch any of this. The only thing I'm going to do in this place is, now I say there's none here, then I come to display. Now in display, if I don't need this square brackets, I just say none. I don't want anything here. You can make your line width to be thicker, or you leave it the way it is. Then the next thing is I come to this place. This is where I'm going to change color. Do you see? I'm going to change the color. Then let me change my color to red. You see, it has changed. So now this guy is red. That's my root string. Please, before I continue, is there anything that anybody wants to ask me? So I will know, because I know I can speak fast sometimes and I can move really fast. Is there anything that is confusing in what I've been doing so far? Nobody's talking to me. Okay, thank you. So now I've been able to give this my root. Then I can have a sequence that is my sequence of interest. So that is what I call the query sequence. Like she told us, I want to give it a color too, so that the person that's looking at what I'm doing will quickly see what my sequence of interest. Then I can also do this. Maybe let's say this is my sequence of interest. OP8726 is my sequence of interest. 1, 3. So let's just do it. You come again to this place. Sub 3, just like I did, I want to give it a different color too. so that somebody can see it. Okay, it's just the same way. Come to display, remove this. I don't want to put anything here except you are lumping them together, then you want to give them a new name. Then I'm changing that one. Let me change it to color yellow now. I say okay. So it changed. You will see. So this is my query. So I can just, in case I'm publishing this, I can just write that my query sequence is in yellow and then my root sequence is in red, and then that's it. So this is what my tree has shown. Then the next thing I want to do is that sometimes when we're working with because we are going to be publishing our work, or because we are going to be writing a thesis, one thing we do is that we already know that it is scientifically correct that scientific name should be italicized. So the next thing I'm going to do is to get my image downloaded. Look at it. Do we see where I click image? So what do I do with that? I said save as. I'm going to be doing two things. I can save as TIFF and get it downloaded like that, and then it's going to be saved. This is not new to us. We all know this. Remember to rename this. Just put on the phylogeny, then an analysis of what's the name, Pseudomonas training. So I'm going to save it. Okay. Now, so that I know the name when I want to check the tree. So I can save it this way. Then, just for my copy, in case I'm working on it, I'm beautifying and make an error, I can look at, okay, where the error is. The another thing is that I can also save it as SVG, this one. That is this. In this case, I'm going to take it to do some other beautify work on it. If I don't want to use iTool, I'll be going to iTool. We'll go to, I will get iTool done. So let me just put it in this place again. Again, the same thing, pseudo training. Okay, so we have this already. Now, so this is what we have for the tree that we've drawn. Now there's something. Now I can now come to this place that say now, okay, this is now. Look, do we see now this is what we need. This is what we need to be able to work with iTool. This is what we need to be able to work with iTool. So then we can just click on this, and then we click plus bootstrap value, everything. If you want the branch net to be there, you click on it. Okay, it's not permitting us. I don't know why, but we can always have a option to click on that. So we just say okay. So then this I can now say, okay, save as. Then you save this in your folder as a new week tree. You give it the name. Now there's something I want to show us that we'll see here. Let me just put this name. Now you see that I say all file is not showing us .new week, so put it as .nwk, so that when you want to work with it, it's going to be easy for you to find it in iTool. That's why I put .nwk, and then I can close this. So that's all. So that is what I'm going to be taking to iTool to work with. That's about that. Now, another thing I want to say is that there are sometimes that you are working with more than one general, and then you want to group them together. Maybe you have a long length. There's a way we can clump them together. So we can just click on those who want to clump together. For example, if I want to clump these two together, then I'll come to this place, I highlight it. I want to clump them into one. Like say select subtree. You come to drop option. Oh, sorry, I'm coming, please. Oh, sorry, I can't find out now. Maybe at long last I remember. I'll say, but we could clump them together so that sometimes you see some tree that they have one, something like a big leaf, and the other will follow. I think I can't recollect how we do that presently, but we could do that as I want to show us. Then also, now I can italize this also from here, like I showed us before. That is also very simple. Select tree, then come back to display. I want to italize it. I remove this, then I go to font, then you come to italize. Now, why this may not be the best approach, because even with your it's going to italize everything, including the strain name that you're not supposed to italize. That's why italizing on MEDA is not the best. If you want to italize, either do it on I2 or you do it on SVG annotation or an SVG folder, something that can open an SVG file. So you can do it in that place. So that's one thing you could do. So now let's go to this is NJ, neighbor joining based phylogeny. Now let's go to the next one, which is maximum likelihood. Let's try maximum likelihood. Just a moment, please. Okay, now go to maximum likelihood. We want to draw a tree, phylogeny, construct with maximum likelihood. I told us this is slow, very, very slow. Now, you see what in those of the model that we have, I don't think we have Tamura. Do we have Tamura in our model? If you remember that model, we have this guy. We have this guy. This guy is the best guy that they told us. Okay, and that's what we are working with. Based on, based on our sequence. I checked it from here, and he told us, he gave a list of what we can work with, the models we can work with, and the best is this guy, is the one that's number one. So then what do we now do? So the next thing we do is, okay, this one. So now we are working with it. Now you will notice that this guy is very slow. You see it's counting. The replicate is counting. It's more, and it is more rigorous statistically. So it takes time. So if you want something that you just want to show to your supervisor quickly, or you want to present in a conference, you can quickly use neighbor joining, and then that's the. But if you want to really be sure of, you want to talk to what you have, the best thing to work with is maximum likelihood. Okay, so let's see this guy as he works. So we are waiting for this guy. While waiting for this guy, okay, we've done this already. Okay. Okay, I've explained all of this. Okay, so this is where we're going to next. Okay, so well, let's go back to that guy and see what he's doing. Okay, you see it's very slow. So now, while we're waiting for this guy, let's try something with iTool. So I'm going to go to iTool, and if you are working with me on our system, we can always go to it. This is the link. It's itool.enb.dev. I think we have to register with them to be able to use it. I don't know, maybe they've changed that now, but I'm sure we have to register with them to be able to use their tool. So let's see what we can do. Now, what I'm going to say is that there are some things I'm really not familiar with iTool, because I often don't use them much, but now I can use it to some extent and I can explain a few things to us. So let's go to my tree. First, I'm going to sign in, okay, to have the optimal use of it. Okay, now I've signed in, okay. I hope you are seeing my screen, please. Are you seeing my screen? Let me know. Can you just point to me if you are seeing my screen? Okay,
yes,
thank you.
Can see your screen.
Thank you very much. So now the next thing is I'm going to look at, it says my tree. I'm going to upload my tree. Remember, we already saved our tree with a name. Do you remember in a new week format? Okay. So let me go to tree upload. Now I'm going to be explaining some things, but I'm not so, okay, there's it. Okay. Okay. Now I am not so familiar with, you see, I want to show something in my tree upload. I want to explain something here. Now you see this, it says give tree a name and tree definition.text. One thing that I want is that what we're supposed to do is that I can just give the tree a name if I want, and then I'm going to be writing here how I want it to arrange my tree for me instead of me doing it manually. I would have shown, let me show something in this place, but I'm not really so familiar with it. That's why I'm not going to be teaching it. Well, I don't teach what I don't really know so much. Now, these are kind of colors. These are the colors we're going to give. This these values are colors that you can just save in text file and then you upload in that place, and with the name of your organism as you have listed them on the tree. So this will give us the color we want to use. But I'm not going to be working with this because we can, instead of doing this automated in an automated manner, we can also do it manually on I2. So that's what I'm going to be showing us. So I will give them different color, but this will make your work faster. You just upload everything together and then it gives you your tree in a beautiful format in the color that you want. But for me, I'm not too familiar with this time. I have used it before, but I don't use it often. So I'm not going to say I'm an expert discussing it. So I'm going to be discussing the other way. So let's continue with I2 now. Okay, here we come. So I'm going to look for the tree I've just placed there. I don't think it's here. So you see, I have so many trees here. So please, okay, it's this guy. So now the next thing I want to do now is that I'm going to click on it. Okay. Now, once I click on it, it's going to give me that tree that I have. You see, it gave me the tree. Now I want to beautify this tree. That's what I want to do. I can make it circular. Do you see it? I can make it rectangular like you see it. It's unrooted, you see. So we can do all of this here. Okay. Do you understand what I'm doing or I'm too fast? Am I too fast? Any response? No, but I'm not too fast. Thank you. Okay. So now the next thing we want to do is that let me work with the rectangular format. And then we want to, for example, I want to italize this. There's something I should have done. Maybe I should have come up with the names of these organisms so that it will be very easy for us. But let me just explain how it works now. You see, the thing is that I want to give them, if I want to give all of these different colors, I can do that. So what I will do is we come to advanced, then I select the node I want to work with, then it said choose color range. Okay. Now this is it. Label. Do you see my label? It's the label. This guy that I want to change the color. This is where I say, do you see the label? Then I say color. I'm changing the color now. Okay. I'm choosing my color. Then the next thing is I want to italize. I've italiced. I want to edit the label. So it's going to tell me to edit the label. Let me call this Pseudomonas aeruginosa. I don't want to use this to explain something to us. Okay. Now it has two names. Now, do you see I've changed the name? Now you will notice something here. Okay. These are all of them has been italiced. But I don't want this because this is not a scientific name. Do you see this guy? This OPS is not a scientific name. So what do? I want to change. I want to remove this. So I come to this again, still highlighting it. Then I come to multi-style. Do you see multi-style here? Hello, are you with me? Okay, thank you. So from my multi-style, I can now change them. So do you see? So the last style is, which one is that guy? So I have, let me check now. Okay. I have three levels. This is 1, 1, 2, 3, and the third one. So this is the third one that I don't want to be italicized. I want to straighten it. So let me see. Let's see if I've done that for me. So it should do that for me. Update label. Close. No, it did not do it. What happened? Okay, let me see. Okay. Okay, now this is it. We can do it here instead of going to that place. Do you see? Edit multi-style, and I selected that guy. So I can do that now. So this is the one I want to edit. This is the guy. So I can just unitalize this one, unitalize all of them first. So I've removed the Italy now. Then I'm going to italize only these two. Then set selection. Okay. Let's go back there. Select, italize, close. Just give me a moment, please. the multi-label. Okay. Customer, please. I want to know what is wrong. Okay, I think it's fine now. And let me go to this guy. Oh, I don't want this. I'm coming, please. I'm trying to see what is happening because okay. This is not italicized. Okay, I'm italicizing this. Okay. I'm digitalizing this. Okay. Let me see. Digitalized. Just give me one, please. I'm trying to see. Okay. Okay. Oh, okay. Let me do it. Okay. Utilized set selection. What's wrong with this guy?
Yeah, maybe not default, yeah.
Yeah, that's not going to I've done that. I've selected it and I've done that. Let's see what happens. It's April. Okay. Okay. Just give me a moment, please. Let's do this multi-level. Utilized. Selection style. Okay, I think we got it. Okay. Okay, now can we see it now? Hello. So let's go back to it. I think I got it now. So we come to this. I've been able to utilize it, and let's work around it together now. We go to label, then we come to edit multi-style. So let's work with another one. Let's start with this. Let's start with this one. Okay. So let's work with this one now so that we can work with that. So edit label. Okay. So we are going to rename this guy now. Let's give it a name. Pseudomonas aeruginosa 2. Pseudomonas. Okay, aeruginosa. Okay, we've given it the new name. So the next thing we have to do now is we want to edit multi-level so that this guy will not be italicized. This is our screen level. So we just come here and then we do italicized. Then we click set selection. So it's done that for us, then we close. So if we want to change the color now, we can change the color in this place to any color that we like. So I've changed it to blue, choose, and then we say close. It's not giving us. Okay, maybe because we did not select it. So let's just change the color. Okay, let's change the color from here to blue. So we can just change the color to blue now from here and choose. Okay, it should give us blue by now. Oh, so don't let's worry about that. So we have this already. So we see that we can now italize this. That's where I2 becomes very important and very useful for us to use, because we could not italize this without italicizing the strain name, that the non-scientific name. That's why I2 is important. Then we can also do so many other things, like maybe if you are going to be talking about the clades, then you can begin to bring the clades together. You can give them a color. Most especially that is done with circular genome map. If I circular phylogeny, if I think circular phylogeny and I want to group a group together, you can also use iTool to be able to do that. I won't be able to recollect that now, but I think I remember I've done that. I've used that before in the past. So you use it to cycle it and say, okay, this particular group belongs to this color. This color is for this particular group, or this color is for this particular clade. We can also do that. Now, let me talk about clade now. Clade is just that the branches that are evolved from the root strain. Okay, so let's check what we have before. I think this is all I can talk about iTool. I'm not too familiar with iTool. I use the, but I'm really not too familiar with it, and you could see the way I was working with it. But just because I have basic idea that we can still work with it, I said, let me discuss. I think we can just work and move on from there. So let's look at the next guy, the other guy we've been working with. So you can see what I said about maximum likelihood. We spent like 15 minutes and it's still on it. And this just only maybe 10 or 11 sequences. Now imagine you have like 100 sequence. You think it's going to take you a lot of time. That's why I said sometimes we are working with a lot of sequence. You have to use other tools, but those tools are terminal based. Like IQ3 is terminal based. It's not what we have that we can download like this and work with it. You have to work on it on Linux, WSL, or a Mac. So that's why when you're working with a lot of data, it's better you work with neighbor joining. We will see that the difference is usually not too much. We will see once it finishes this. So that's about that on phylogeny. Let this guy finish, then we can look at what it is, how they look like. Then I can entertain some questions that you have for me. Like I said, I don't think the time allotted to me for this will be enough. And this almost 2 o'clock already. I don't know what time is it in that place. Maybe 8 o'clock already or 7 o'clock. And so I don't know. So that's why I said for those 3 things, the time may not be enough for me to be able to explain. So let's see what this is doing. Then we can move to submitting our sequence. Let's see what this one will have for us. So while we're waiting for that, I think there's any question for me, we can just give it to me in the chat so I can respond to them. Any question for me? Oh, which part is that, please? Somebody said, which step do you want me to, somebody should repeat a step, please. Which step are you referring to? Can you please type here so I can go across it again? Or have I done that? Ah, I'm really sorry. Are we still with me? Okay, okay. I'm going to do that now. Just 1 minute. Now, somebody said I should share a particular. What data do you want me to share? Which one is that? Is it the sequence I sent? Okay, the sequence. Okay, let me see if I still have it. Just a moment, please. Okay, now there's something because I'm kind of rushing. There's something I really also did not do. Now, you see, you remember that when I downloaded those files, I went to use them straight. One thing we're supposed to do is this, just to include our query sequence, because sometimes what you download is not what you have downloaded is not your query sequence is not part of it, because your query sequence is your own sequence. So you have to add it to it. So let me quickly do that. For example, this is my query sequence. So for me to have, where's that guy? Just a moment, please. Okay, I'm going to open that my FileStar file. Okay, which I call training.faster. Where is it? Okay, there's it. So what I'm going to do is that now, because I was in a hurry to jump to what I was doing, now I'm going to include my own sequence here. So let me just copy it, which is my query sequence. This is it. This is where we have it. So let me just open this, copy it, okay, then add it to that sequence to his colleagues. I think it's this one. So this is my query sequence, and then with this I can now do my normal analysis. What I did was with what we have. Another thing I want to show us, please, while we are thinking of the question we want to ask me, okay, is this, and while we're waiting for that, now we can edit this before we put it. Now you see, I didn't see those names because I didn't edit this. So you can just edit this straight. You already know what they are. You don't need to put 16 years. You know it's 16 years you are working with. And the next thing you do is please just put an hyphen. It's because there are no hyphen in between them that I could not recognize the names. And then that's why I don't have all of those names shown in that place. So if you do that for all of this, then you will see that it's going to show the names when you go to Mega. Okay, so I'm going to do that now. I'm going to complete with this 10. Do that for all of these 10, then we are going to open in Mega again. Just a moment, please. So when I open Mega, you see that it will show me all of this name and the session number that we only saw before. Okay. So I will use this to explain again how I open it in Mega. Just a moment, please. Hello, please. Can I still go with your time? I think I have 1 minute left. The coordinators. Nobody's talking to me. Can we hear me?
Yes, sir, we can hear you. You can take 5 minutes extra to
Okay,
just
5 minutes. But that means I will not be able to show how to submit to GeneBank. Maybe some other time we can do that. But
yes,
it's not
sir.
okay. Thank you very much. I know when I saw it, that's why I said that minute given to me will not be enough to do all of those three. Okay, just a moment.
I think if you use replace space by hyphen, it's easier.
What do you say?
Replace, use find and replace.
Define? Find and replace. Find and replace is very difficult for me because you see what I'm cleaning is not together. So that's why I want to do it manually. So if I do find and replace, I won't be able to do everything. So I will still do what I'm doing now. Do you understand what I'm saying? That's why I'm not defining. Oh my God. So that's I'm not doing find and replace. I would have done find and replace because find and replace will not do that for me. That's why I'm, except I use find and replace to remove all of this. Okay, let me just do this. Control H.
Find space and replace with I. No, just only space.
Okay, so now, okay, thank you. So now I can, where am I?
Okay. Yeah, there is still just hit bar, just only space. Space. Just only space, bar space.
It's not working with me. Do you see? I've done what you asked me to do, but did not change it. So let's just do, soon be true, just in a moment, please. It's just 10 sequences that we have here. So we can get around it. Okay, let me try this again. Let me see. Okay, I think it worked. Thank you very much.
Yeah,
that worked. Okay, yeah. Thank you very much. That worked. Okay, so we don't know this. We already know the name of this one. Okay, so let's say Control S. Now I'm going to open. Oh, I said I'll be given 5 minutes and I'm going to round off. Okay, so I'm going to my Mega again to open this my new file. Okay, let me see. I think it's ready. Okay, it's ready. So let's look at it. Do we see that there's virtually no difference between what I have with, if you have that, let me see if I can open that. Let me see if I can find that open before I continue, before I round off. Oh, where's this guy? Okay. This is one. This is the one I did, and then this is the other one. So you see that this is, can you see this OP872619 they are together. So let's see that it's almost that there are no difference between them. Do you see now? So that's what I said, that if you want to quickly show something, there's virtually no difference when there are no complexities. Only when there are complexities that maximum likelihood is going to show you some differences. That's what I said earlier. Now let's do what I was asked, that how I opened it, that somebody says it's not clear. Then I think at that point I'm going to stop. Okay, now let's close this one. No, I don't want to have this discard result. Okay, so let's come to this guy. Open, do you see my file? Open a file session. Then I click on it, it will get me to the folder where I'm going to work with. I think it's this folder now. Then let's open it. So it's open. It said, I have a file already activated. Do I want to use another one? Yes, I'm using another one. I don't want to save anything here. Close without saving. Then I can work with my new file. Let me close this. Discard result. Close this. Just a moment, please, because it not opened out for us. Okay. Let's close this too. Now, let's start again. Open a file session. This guy. Open. Align. Okay. Now we have it. So this is what I said now. Can you see that the names are shown? Okay. So, is somebody talking to me there?
Yeah. I was listening.
So can we see the name are shown now? Just because I put that hyphen. If it's not there, it's only going to give me the first lines. Okay, so let me close it and let me do it again. I got there. File, open a file session where I've stored my file. Click on it, click open, click align. Then I'm going to work here. The next thing I do is to do my alignment, Control A, then align. You can choose muzzle or cluster W, but don't use codons, please. Use either align with cluster muzzle or align with muzzle. Align with cluster W or by muzzle. Don't use codons. Then click on cluster W. Okay. Okay. So then we have this already. Then the next thing we do is what? We come to data. Do you see where is data? They say what? Phylogenetic analysis. Then you click yes. Okay. Then from there, we come to this place. We already did this before. We know the model we're going to use. If you are not using maximum likelihood, model is not important. But if you are using maximum likelihood, it's better you check the models from here and it gives you the best. The best model will be up. You can click on it, or you can click on use all data, so that that will automatically select for you without you testing the model. So let's go to the next one. They say phylogeny, construct with neighbor joining, and let's go there. It's just, you see, it's not giving me the model. Now you can also arrange this. You can reduce this if you want. You can say 500 test of phylogeny. You can say 100 for most of them are working with eukaryotes, fungi, or other microbes. You can reduce the bootstrap replicate value to like 100 because of the size, the length of those sequences, and then the variation that is commonly between them. So you can also reduce it, and that's all. So that is all I have to teach. I'm sorry that I took more than the time allotted to me. It's just that what you asked me to talk about is more. Thank you very much. I'm grateful. Thank you for the opportunity. Thank you for attending my class. I'm sorry I speak too fast. Thank you very much for everyone that be. And thank you, Afro Bioinformatics Link. Thank you very much for the privilege. I'm so grateful for the opportunity. Thank you so much.
Thank you so much, sir. Can we keep clapping with our digital hands? And let's appreciate him. He spent over an hour, about 1 hour and 20 minutes, almost 20 minutes or so. Well done, sir. We really appreciate your time. I don't know, someone is raising his hand. Is there something you want to say? Kindly unmute and okay, drop your hands. All right. Do we have any questions that you want to ask or and has not been asked or has not been addressed? Do we have anyone with questions? All right,
I
appears
see.
there is
I think I
none.
see a question here. I think I see a question
Okay.
that I may want to talk to, but I may not really get what the person is trying to ask me. He said there are values associated with the clades and the trees. How do we relate it with the range value as we in map reading? It is okay, I understand. Now I think I mentioned it. You see, there are different, it depends on what you are working with. The most important is the bootstrap repeat value, which are those ones that you see at the end of the tree. You will see them. Let me see if I still have the tree. I have the tree drawn now. Okay. Now can you see my screen? Sorry, I stopped sharing. Just a moment, please.
I don't. It is not present.
Okay. Okay. Wait. Okay, I'm coming. Can we see my screen now?
Yes, sir.
Okay, so I'm asking that question now. Look at this. This is only showing us how close they are to one another. What this says is that, you see what I said? I said the bootstrap test of phylogeny, which is the bootstrap repeat, is saying that set round 1,000 times and keep grouping them together. Keep grouping them together. These two guys will appear together. At what percentage? Do you get what I'm saying now? This go get at what percentage? Okay, from 100 to 50 is okay. Anything less than 50 shows that grouping is not, we can't trust it. That's what we call it test of phylogeny. Do you get it? So we can't trust it because it's just that means that at every time I search, I could mix them up randomly only less than 50 times. But for 100 times I could mix. So that's what those numbers refer to. So it tells you how reliable is they are coming together. So I think if the person asks the question, I want to be sure if I've answered that question, please.
Do you mean a sort of simulation, number of simulations?
Yes, something. Okay, let me use that word for those who use simulations. Okay, number of how reliable is the simulation. It is percentage reliability of your simulation. That's what it's saying.
Okay, please, instead of using percentage, there are sometimes in phylogenetic tree, values are just shown 0.5, 1.0 instead of percent. Is it also okay to use such?
No. This one you're asking me is another thing. There's something they call the branch length. The branch length value is just the thickness of the length. That's what they say. That's one that you used to see on there, say 0.05 or 0.01. That has not to do. It's just only telling you the length of the tree, the branch length of the tree, the average is what it's telling you. If that one is not talking about reliability of your phylogeny or of your tree. That is, it's just the scale. Let me use the scale of drawing. Do you understand it now?
Yes, thank you.
So it's just scale of drawing. It has nothing to do with what is happening in your phylogeny or not. It's just a scale of drawing. So that's it. So any other question for me? Okay. So thank you.